Now I'm going to share my screen and let's take a look through the quiz. Some people still aren't here, but they're lost. All right. Can you see it?
Yeah, we can see it.
Great. Okay. So somebody walk me through polarized growth.
Isn't polarized growth when all of the growth inside, I guess, maybe the hyphae or wherever it is, it's all centrated all in one exact spot rather than being dispersed? At least that's what I got from reading the article.
That's it, exactly. Okay. So how is that different? Oh, I got an echo. How is that different from other epistles?
I guess most opisthecans undergo cellular growth through mitosis, which basically is just spreading of the cellular growth, and it can happen at different sites, not just in one spot.
Okay, you've got a slight misconception about mitosis.
Okay.
Mitosis has nothing to do with cellular growth.
Are we talking about like the growth of a cell or like getting more cells?
It's just the nuclei. And fungi do mitosis quite a bit. So it's just nuclei.
I see what you're saying. Okay.
Yeah. But yeah, the fungal cells tend to only grow at one point, and that's what gives it that kind of spaghetti-like structure, I suppose. Okay. Other pisciconts, so all the animals, right? Our cells don't exactly do that. They're swelling up all around as they grow. Spitzenkörper, what is it? How does it relate to growth direction? So this kind of looks at polarized growth again.
Isn't it a spherical or aggregation of, what did I put here? Secretory vesicles?
Yeah.
Isn't it the way it grows, right?
What are the vesicles doing, I guess?
They're reacting to light, right?
In some species, yeah.
So I guess not necessarily just light, but they're reacting to different environmental triggers.
Yeah, so it's
direction.
If you've got like your hyphal cell right here, the Spitzenkörper can move around inside there. It tends to stay at the very tip though, just like half a micrometer back from the bleeding edge of the growing hypha, but it can move around to the side. If it moves to the side, what's going to happen?
It's going to grow out
that direction.
It's going to grow out the side. And in fact, the Spitzenkörper can even split. And so the fungus can branch in that way. Okay. But it's definitely the region, it's a bunch of vesicles packed together, secretory vesicles. What are they secreting?
Yeah, like digestive enzymes.
Digestive enzymes. And so this is stuff that's either digesting the substrate that the fungus is growing through, or it's actually helping to soften, to partially digest the fungus's own cell wall. Okay. So that's what a Spitzenkörper is. Question number 3, has it evolved independently multiple times, or was the Spitzenkörper lost in most zygomycetes?
It's still inconclusive.
We don't know.
Great, yeah, that was a trick question. I actually just copied and pasted it. Okay. What new information did the Fisher paper present that contradicts what we read in the textbook?
Okay, I'm hoping that there's more species outside of higher fungi that have Spitzenkörper.
That's
Is
it.
that it? Okay.
Yep. Our textbook is a bit dated. There's a newer version, but it's like $250. So, okay, whatever. That's what papers are for. It goes ahead and says that Spitzenkerpers aren't found in zygomycetes. That's what our textbook says, which was true at the time. How many do we have now?
Isn't there 3 cases that they know of that are outside of it?
3 so far, according to the paper. Yep. Which kind of does bring up this question: has it evolved multiple times independently, or was it lost? Before we saw the Spitzenkerper in any zygomycetes, it was relatively safe thing to say, like, oh, it just evolved this one time in Dicaria. But now we're not so sure. Okay, mycelial strand versus rhizomorph.
So mycelial strand is basically undifferentiated hyphae, while rhizomorph is more differentiated, it more acts like an organ. Basically.
Yeah, acts more like an organ. Cool. 3 types of fungal spores. There were more than 3, so everybody give me one.
One of them that I did was the sporangiospore that we talked about for
Okay.
zygomyces. So haploid sporangiospores.
All right.
An amerospore.
A what?
An amerospore.
Okay. What's an amerospore?
A single-celled fungi, I guess.
Amero just refers to like whole or entire. So I got 2 drawings for you. I don't know if you can see that. All right.
Yeah, we can see it.
Great. The one with the lines through it is not whole. It's kind of like divided up, split up. The solid one is an amerospore. So you'll see that term sometimes.
Is there a name for the one that's split?
Phragmospore, because it's fragmented.
How about a zoospore?
Zoospore, yep, we've seen those.
Yeah.
Those are just in the paraphyletic chytrids.
Yeah.
What about the ascospores, or how do you say those?
Ascospore, yep.
Okay.
What? Tell me about that.
Aren't they the largest group of fungi?
Ascomycetes are the largest group of fungi, but not all of their spores are ascospores. What makes an ascospore an ascospore? Can you repeat that? It was kind of quiet.
They are meiospores, so they reproduce through meiosis.
Yes. Specifically, in keeping with the tradition of we name spores after where they came from, ascospores are made in an ascus, and they are the sexually produced spores through meiosis.
Quick
All right.
question.
Yeah.
Which type of spore is the one that you showed shooting out? Out of like cow dung?
Sporangiospore.
Okay, those are sporangiospores. Okay.
Mm-hmm. That thing that shoots out is actually pretty large. If you hold your face over some cow dung, not that you'd want to do this, but it's kind of fun. You can feel them like hitting you in the face. They shoot off pretty hard, and they've got a lot of mass. It's actually thousands and thousands of spores in a mass, and the thing that's being shot off is the sporangium. So it's a bit counterintuitive that way.
And that's fun.
It is fun. I mean,
Okay, all right.
Yeah. I mean, try it out. Go to a nice fresh cow pie after some rain, and don't breathe in, but stick your face real close, and you can feel it.
I just have to take your word for it.
No, you should try it. It's kind of fun. And they do stick to you, so you can grow them. All right. Any other spores you got questions about? There were quite a few like spore names that dropped on you.
How about that all spore, something like a spore that doesn't move? Is that what it was?
The oospore?
Yeah.
Okay. That's not actually from a fungus. That's from an oomycete. They are stromatophiles.
Okay.
They look like fungi when they're growing. They've got filaments and everything. And so traditionally, mycologists have studied those. I'm choosing not to really deal with them in this class because they're protists, not fungi.
Okay. No fungi. Okay.
Yep.
Thank you.
All right, what about this spore?
Zygospore.
Zygospore. And that's the zygomycete analog to an ascospore, right? This is produced by sexual reproduction. Meiosis takes place inside of it. All right. Zygomycete sporangiospores are what?
Dr. Zahn?
Haploid.
Yes.
Would the picture above be I can't remember the proper name from it, but when you were talking on Tuesday, the one that is like does it itself versus needing two separate sexes?
Homothallic.
Yeah, homothallic.
Yeah. Yep, it's homothallic. Even though homothallic zygomycetes are relatively rare, you see them turn up in like every illustration. It's hard to find pictures of heterothallic ones that are drawn because this is just a convenient way to draw things. Yeah.
Awesome.
If I put zygosporangium on that one, would that work?
Sure. Yeah, I'll take it.
But technically it's a zygospore.
It's a zygospore on suspensors. Yeah.
Oh, okay, so it's just a single spore. Cool.
Mm-hmm.
I think in the book it actually labels it as zygosporangium as under the picture.
Yeah.
But I also put the zygospore as well. So
yeah, I'll take it. It's fine. All right, sporangiospores and zygomycetes are haploid.
Haploid.
Yep. Okay, cenocytic.
They're aseptate.
Yep, one big cell. That's what cenocytic means, the big shared cytoplasm. Okay. So no septae. And what two families did I have you read about in the Webster and Weber book in the Mucorales clade?
was one of them Mucor?
Mucor.
And Pilobolus something.
Yep.
Pilobacea or something.
Yeah, often pronounced Pilobolus.
Pilobolus, thank you.
Mm-hmm. I don't think there's a wrong way to say it.
No, it helps.
That's how you'll hear it most often.
Awesome, thank you.
Mm-hmm. Okay.
Hey,
Question?
Dr.
Yes.
Zahn?
Mm-hmm.
So when I was reading those last night, obviously they're pretty dense, but for question 10, was there, I mean, that seemed like it was an overarching theme, but for me, when I was looking through it seemed like there were so many different classifications that I had a really hard time figuring out which ones were the main ones.
Okay, so let me show you in the text here. It was 7.3. Okay, examples of Mucorales. You can see here the two ones, the Mucoraceae and the Pilobaceae. So if you read 7.3.1 and 2, which was the assigned reading, if I'm not mistaken, yep, 7.3.1 and 7.3.2, that was the Mucor and the Pilobolus. So if you gave me Hilaria, then I'll take that too. I'll take any of these in these two main groups. Does that help?
Okay, I think I was just second guessing myself.
Okay.
Thank you.
Yeah. I had you read about the Mucorales and not all these other crazy groups. Remember, the two main groups of zygomycetes are the Mucorales and the Zoopagales, which contains a bunch of groups in it. But mucors are by far the most common, most abundant, and have the most species. And so that's why I wanted you to see those, because that's what you're going to encounter kind of in daily life. That's your bread mold stuff. That's your cow pie stuff. You don't have to go cutting open an insect and dissecting it and hoping it's got microscopic things. So that's why I went with those. Okay. All right, questions about the quiz?
Would you mind just briefly going over 1 and 2 again?
Sure.
Thank you.
Okay, so let me do a little drawing here. So here's a fungal hypha, and the Spitzenkerper is that cluster of tightly aggregated secretory vessels. All right, so they're excreting a bunch of stuff. That stuff is helping digest things in the environment. It's like if this is growing through wood, it has to physically get through it, and so it's digesting the wood, but it's also kind of digesting the tip of the hypha. If that secretory body kind of moves around, that means the direction of the fungus growth is going to change. Okay. That's because the fungus can't really grow very well unless it digests the tip of itself, okay, because it's got like a cell wall, and so it has to soften that cell wall in order to push forward and lay new stuff down. And so the Spitzenkerper is found in ascomycetes and basidiomycetes, but also now we know it's in a few zygomycetes as well. And it's kind of controlling the direction of growth for that hypha. Okay? And an organism, a cell that grows in one direction and on one side and not the other, is said to be polarized. And so that's what polar growth is. That clear it up for you?
Yeah, I just put that polarized growth is when the microtubules extend the cell wall to either ends or poles of the cell.
Sure.
And other epistoconts are monopolar, growing towards one end.
No, the fungi are definitely growing at one end. All right, does that make sense?
Okay, yep.
Yeah. So an animal cell starts out kind of small, right? So it splits off after mitosis, and then it'll actually just grow outward. The fungal cell only grows at one point of the cell, and so it makes a tube as it keeps growing. All
So,
right.
Dr. Rob, could you say that polarized growth is basically directional and organized growth?
Yeah, I mean, think about like a tree. This isn't happening at the cellular level, but at the macro scale, trees have apical meristems. Remember those?
Yeah.
Yeah, and so think of it's not an exact analogy, but the Spitzenkerper is kind of like an apical meristem, actually. So it's only growing at one end. The tree itself also has secondary growth and all that, but the tree isn't extending from the middle, it's only growing up from the top. Same thing for fungi. As a matter of fact, if you chop off the very tip of a growing hypha, it will kind of seal itself off, and a new Spitzenkerper will form in a cell back behind it and start growing. So it almost works like apical dominance in plants. Yeah.
Dr. Zahn.
Mm-hmm.
Can I ask a question about something you were just talking about with the Spitzenkerper? It's technically from the paper, if that's okay.
Sure.
So,
bring up the paper.
Yeah.
Uh,
And it's during the discussion portion of the paper.
Okay.
It's the second paragraph. It's talking about how the Spitzenkerper changes its position within the apex, and that changes the direction of growth. So in the paper, they continue to say that additional investigation is warranted on this issue.
Yes.
So I'm just wondering, does that mean we don't know what causes the Spitzenkerper to change its position? Like, is it environmental cues that does it? Is that what they're saying?
They're saying we don't know.
Okay.
Yeah. It's not too hard to observe it with a microscope, and we see that it does change sometimes, and then it starts growing that direction. So it's definitely a strong correlation. Maybe it's safer to say we haven't proved beyond doubt that the Spitzenkerper is actually responsible for the growth, but evidence is real strong. We know that it's sending out digestive enzymes that weaken the cell wall. We can see the bits of cell wall being put into place, scaffolded up as it grows. We've actually tested how soft it is with microscopic needles and weights, all that. And so we've kind of like prodded along the fungal hypha and seen that where the Spitzenkerper is, it's a lot softer, and then it starts kind of laying down cross-links, sort of like peptidoglycan in bacteria, right? Chitin isn't firm until it's been cross-linked, and with the little glucan proteins and whatnot. So we kind of know a decent amount about how the Spitzenkerper is operating, but we don't know everything. And so when something like that winds up in a discussion section of a paper, when I read that, I think, oh, reviewer number 2 works on Spitzenkerper growth and wants them to say something about it. So it got put into the paper.
Okay, that makes sense.
Yeah.
Thank you.
Dr. Zahn?
Yes.
I had one more question about the Spitzenkerper too. I was kind of confused between what the difference is between microvesicles and the chitosomes, or if they're almost the same thing.
Off the top of my head, I don't know much.
Okay. Well, that makes 2 of us. Thank you.
Okay, yeah. I'll find out for you and post an answer here.
Thank you.
Yep.
I have a question about like coincidic, like the mycelium. So what's the advantage of a mycelium being coincidic?
Cenocytic is the pronunciation, but advantage is it can grow faster and it can do that cytoplasmic streaming longer distances and have a bunch of nuclei in a given spot. And since nuclei are responsible for, so much of cell growth, they control so much about what proteins are being made and all that. If you have a bunch of nuclei somewhere, it can be growing really fast. And so these fungi can be really fast growers and kind of make it through a substrate, out competing a lot of other fungi, even higher fungi. I kind of don't like that word higher fungi, but yeah. So that's something, I guess, but the trade, and they're doing pretty well. The trade-offs being that it's harder to compartmentalize if part of you is damaged, right? A lot more of your cytoplasm gushes out if the cell gets disrupted.
Okay, thank you.
Mm-hmm.
So in the paper, it was talking about how they were like plunge frozen. Like, why exactly do they do that?
Oh, for the microscopy?
Yeah.
So that's for this transmission electron microscopy. This is actually a really hard technique. You have to embed your cell of interest into like a weird plasticky kind of matrix and then shave it very thin with a diamond razor blade, essentially. And we're talking very thin enough for some electrons to get through. And so that's what we're actually seeing right here. And so one of the processes they do to kind of embed this is that flash freezing. The details of how to do transmission electron microscopy are kind of getting lost, I'm sorry to say. It's not a technique that's used a bunch anymore as people move more towards molecular biology. And so some of the people doing it are like in danger of retiring. My own grad school advisor did this stuff. He never taught me how because he was lazy, and then he retired. And I guess I'm okay with it because it looks like a real pain in the butt, but it's just one of those like weird detail steps that they mentioned.
Okay, thank you.
I have a question as well, Dr. Zahn.
Okay.
So in the paper, it talks about the 3 regions and how it's organized, type of growth, the hypogrowth. Is that characteristic to zygomycetes, or is that typical of most fungal growth?
It's typical of most fungal growth, and especially those with Spitzenkerper. And so they're kind of trying to show that not only does this particular zygomycete have a Spitzenkerper like the Dicaria,
But also all the
it also is kind of organized into these regions of like relative softness and growth and all that kind of stuff. Yeah.
Okay. Well, thank you.
Mm-hmm.
All right. I had a couple questions about the reading.
Great.
So
Paper
just to be
or text?
The text, the book.
Okay. Let me bring it up.
Just to be clear that I'm not doing any extra work, when you say something like 1.2, you mean 1.2 in all the subsections, correct?
Yeah, I'm sorry, I should clear that up. So if I say 1.2, that means all of this. Okay. If I say 1.2.1 and 1.2.3, it means just those.
All right.
Yeah.
So, can you help clarify? I think it's pronounced sclerotia.
Sclerotia.
Sclerotia. So the first question I have about it is I'm kind of confused. Are those different species, or are they just like a segment of a fungi?
A sclerotium is not even really a growth form. It's a resting phase for a fungus. And a lot of different fungi are capable of making sclerotia. So it's like saying a rhizomorph or a hyphal aggregate. It's just another one of those like kind of fungal growth patterns, organizational structure. A sclerotium tends to have a hard coating, and so it acts kind of like an organ almost. There's some cellular differentiation. It's a way for a fungus to survive dormant for long periods of time in harsh conditions. You'll often find them in Arctic fungi and in desert fungi for obvious reasons.
Okay. And to continue on with that, they used the naming, like the Latin naming system for them. Is that just to specify the types based off of a fungal species, or that's where I kind of got confused because there was the
you see what section?
Yeah, let me go back
okay
to that. So it was 1.3.3.
All right, so these Latin names that you're seeing here, like that? Yeah, these are just, they're telling you stories about different species that form sclerotia.
Okay.
Yeah. So they're saying here, like, this particular species, Paloporus, has subterranean sclerotia that can reach the size of a football. Yeah.
Awesome, thanks.
Let me see if I can find a good picture of sclerotia. This is what I'm thinking of here, D in this photo. You'll see these often on like grasses. Okay, it's just kind of this hard, dormant fungal state. Here, this is, I'm thinking ergot. A, C, D, E. Yep, Claviceps purpurea. So that's ergot, famous for poisoning people. It's full of hallucinogens. It grows on rye. And so in the 16 and 1700s, people had a lot of rye growing in New England. This would wind up on some of their plants. They'd grind it up into flour, make their bread, and then they'd go crazy and their limbs would fall off. That's a sclerotium meant for overwintering. And so, yeah, it's just pointing to some different fungi that make sclerotia and saying, hey, here's how this one does it. Yeah.
Okay, thanks.
Mm-hmm. Let's see. So, on these major topics that we've covered in our lesson and the reading, do you know them? Do you have questions about them? Did they get brought up in the reading? They did. And there's something that's not clear for you.
Yeah, I explained how physics constrains morphology on very small scales.
Okay. I had one slide on that was pointing at surface to volume ratios, right? And efficiency, if you remember.
Oh, is that talking about how they spread out in a circle because it's the most efficient way to absorb nutrients?
Yeah.
Okay.
Yeah. And it also points to why there are so many things at the microscopic scale that are filamentous. It's because it's physiologically, thermodynamically efficient way of getting nutrients if you're a lysotroph.
And then another question. So for fungi that don't have the Spitzenkorper, I kind of mentioned it in the paper, something about them having like a crescent part.
Yeah.
What's going on with that?
Apical vesicular crescent, I think, is what the paper referred to it as. So instead of the highly organized, dense cluster of secretory vesicles, there's, and they didn't provide a picture of it because they just assumed everyone kind of knows what they're talking about. It's more like that. So it's kind of a little crescent moon shape, and it's not as organized, but the point here is that there's still some region of the fungus that's growing more rapidly than the rest, and that's right at the tip. And so all the fungi that don't have a Spitzenkorper that's like really well defined and organized and has solid structure and it's dense, the other fungi still have their way of doing apical growth. Yeah.
So do those,
sorry, go ahead.
Do those crescents direct growth then? Like
yeah, that's gonna have the same thing. Yeah, like direction.
They don't move around in the same way, or they're harder to see, and so we haven't seen them moving around in the same way. A Spitzenkorper is nice in that it's really clear on a microscope, and so we can like make movies of it moving around. This like AVC, this crescent, is less of a structure, and it's easier to identify like when you stain certain proteins like actin or something like that. I believe yeah, here we go. Okay, so in this YouTube video, this is linked, they've stained actin. And so actin is responsible for like cell elongation, growth, moving stuff around in the cell. And so you can see it's nice and concentrated up at the tips, right? Is this a zygomycete or not?
The answer would be probably not.
Why?
Because there's very few species of Zygomycetes that have Spitzenkorper.
Do you see a Spitzenkorper?
Oh, wait. Yeah, doesn't the end of it have a bunch of actin? So, or we.
But that's not the Spitzenkorper. A Spitzenkorper is like definitely a little marble shape. Like it looks like a little ball rolling around. Since we've stained actin in here, that's not actually going to show the Spitzenkorper. It's going to show activity kind of within the cell. But the giveaway here, look at these little bright lines. Septa.
Yeah.
So that's not an Ascomycete.
So I have one question about the video.
Okay.
So when they come together, do they actually merge and kind of become like one, I guess, root? I don't know how to explain that. Like they come up from two different things and then they kind of like merge together in places.
So the answer is they can. They tend to only do that for sexual reproduction, though.
Okay.
They're happier being just really close together and not actually joining themselves.
It just looked like at the first of the video that some of them they actually went together. So I was just curious if they did that often or if that was just kind of
looks like they did.
Okay.
If you see up close, like.
Oh yeah. Okay.
Yeah. All right, there's your Spitzenkorper. Can you see it?
The very bright dot at the end.
That very bright dot right at the end, absolutely. All right, and you can also see that it's taking a while before it's laying down its septa. Right? It likes to have that cytoplasmic streaming because, I mean, nutrients, it's at the cutting edge of the fresh food, right? It's growing, it needs lots of resources, and it's sending back a lot of resources. And so before it lays down septa, you have to have a nucleus inside each one of those little segments. We'll see that. So this kind of gets at the previous question about what's the benefit of being cenocytic. Even the higher fungi do it for a little while, right? They do it for as long as they can kind of get away with it because it's pretty efficient in terms of moving stuff around the cell.
Is there a way that can change the direction? Because I see that's normally just going straight. Is there a way that I can like turn and do any circles or?
Yeah, and they do. I mean, it's in response to environmental cues. So some fungi are light sensitive like that.
Oh, okay.
Some respond to CO2 gradients. Some actually have a version of like feeling and can notice little gaps in spots. Let me see if I can.
Is it kind of like the same thing as trees? Because I know trees do that with like light or some that will like grow on buildings because by touch is like the same kind of thing.
Yeah, by touch, fungi definitely have a system in place for that, like a thigmotropic system where they can actually feel bumps. And so here is a fungus growing into a leaf. It can actually feel where there's like a little two ridges, and then it starts growing down in there.
The Spitzenkorper will kind of feel that and then change where it is placed, which then causes the growth to follow.
Which causes the growth to follow, as best we know. Yeah.
Okay.
Yeah.
Cool.
There was one picture. I thought I could find it easily, but some researchers actually were wondering whether fungi, how they knew when they were on a leaf stoma, because they like to grow into the stomata, okay, because it's easy to get into the leaf that way. And they were working on this for years, and they couldn't find a chemical signal that they could replicate from the plant that would tell the fungus to start growing down. And so they tricked it out. They just started growing them on like little sheets of graphene like this, basically. And the way to get it to go down was to put two little bumps a few microns apart, and the fungus would be growing over it. It would do the little speed bump. As soon as it hit that second speed bump, it would stop growing, and the Spitzenkerper would move in there, and it would try to grow down into the graphene, which didn't do it any good. But what that told them was that it wasn't a chemical signal from the plant, which we thought was what was directing the Spitzenkerper. It was the fungus noticing that it went over two humps, which is weird. Still don't know how it exactly knows that it went over two humps, but it does. Fungi are freaky, understudied, for sure.
Dr. Stone, I have a couple questions about the paper.
All right.
So first off, it asked the question right at the end if it evolved independently or
Right.
and separate times and stuff, and the answer was it's inconclusive. But does the evidence in having those three species of zygomycetes, does that lean one way or the other based on like theories?
I bet you get different answers depending on who you talk to. For me, I feel like I probably don't know enough about it to make a bold statement. I could see it evolving independently from that apical, like vesicular crescent, right? Because all these fungi have at least that, and all you got to do is organize that a bit more. And so I can see it evolving repeatedly, a few times, but I can also see it being lost in a bunch of other groups because this looser structure was good enough. These groups, like most of the zygomycetes that don't have a clear Spitzenkerper, do a lot of things that are quote unquote good enough that other fungi may look at and go, huh? Right? You don't have cell walls between your nuclei. So
I guess then that leads me to the question of, at the end it says that the SPK is better than the AVC or the crescent.
Yeah.
So why wouldn't it be in more then?
I think this word there I've highlighted suggests is actually doing a lot of heavy lifting in that sentence. They're saying that roughly 95% of fungi on Earth have a Spitzenkerper, and so it's probably safe to assume that it's worth having one. Does that make sense?
Yeah. And then, so as far as the homophthalic and heterophthalic, are both still considered sexual, or is the homophthalic asexual?
They're still sexual because they have gametes. It's like a hermaphrodite fungus. Yeah. There are animals that do that also. Snails can have sex with themselves. Slugs can. And meiosis happens, crossing over happens, and you don't get a clone. And so it is sexual.
But for the homophthalic, then would that be haploid? It only has one copy.
Well, it goes diploid briefly right before meiosis. So in the zygospore, it does go diploid, then quickly does meiosis, and then it's haploid. But that's the result of all sex eventually, right?
Okay, and then just one last thing quickly about the bread mold experiment. Does it matter if we take the picture through the microscope before or after we culture it on the agar?
No, as long as if you can see it, then that's fine. It's just harder to get clear pictures on the bread, I found. Yeah.
Yeah.
I want you to have the culture no matter what, but if you get clear pictures on the bread, that's fine.
Okay, thanks.
Okay. Mm-hmm.
So what you're expecting in that picture is a zygospore, right?
No. Unless you set it up, with two separate growth regions on different sides of the bread where they meet in the middle and have sex, you're not going to see zygospores. I would expect to see lots of hyphae, lots of nice cenocytic hyphae, and I have a decent expectation that you'll find sporangiospores, but not zygospores.
Okay, so we're more looking for like a net of hyphae.
Yeah.
Okay.
And if you've got spores going, I definitely want a picture of those because they're more interesting than hyphae. If you can pluck off one of the little stalks, the sporangia, stick it on a slide, you'll get some pretty cool pictures. Wanted to show you another part of this YouTube video here. And this kind of gets at the paper as well, something that it went into. No, sorry, the book. So sporangium development. This is what you're going to see on your bread after it's been going for long enough and starts getting little black spots on it. You can see here that we're talking like 8 hours from no sporangia to fully developed, ready to go. And it's interesting, it talks about in the book like how this twists one direction as it's growing and then twists the other way. And then as the sporangium itself is developing, there's no actual vertical growth of these things. So this video does a good job of actually showing that in action. So this is bread mold, mucor, zygomycete. You can see the sporangia growing here in time lapse. All right, so they get a certain height, then they start developing their spores. The spore mass starts turning dark, and then they begin growth again. Okay. So you should, with a pair of tweezers, be able to pluck one of these stalks, these sporangiophores containing a sporangium, off of your bread, get it onto a microscope slide, and get photos of that. It may be squashed a bit, but it looks pretty cool under there, and you can see individual spores.
Were we supposed to get slides in that little package that we picked up at the office?
Yep, it was in the Petri dish, the big Petri dish that was kind of taped closed.
Okay. So, because I'm pretty sure I just got pollen in mine. Didn't get any slides in that Petri dish, but I can double check.
It's possible. I double checked them all, so it should be in there. If you didn't, for some reason, go ahead and steal a slide from some lab on campus, or you can use the slide that comes with the pocket microscope. It's just not ideal. It's this plastic one. It does okay. But really what you need is a cover slip to make this functional. Light doesn't go through it as well as the glass, which is why I wanted you to have a glass slide.
I mean, I could just probably stop by like a microbiome lab or something and ask them for a couple.
Yeah, you can do that if you're on campus. Sorry about that.
I have a question about the pollen.
Okay.
Example. So you said that we're supposed to find some dirty water or whatever from a lake or a pond or whatever, right?
Mm-hmm.
And then fill it up maybe halfway. Is that right?
Yep.
And then are we supposed to crack that lid and let air get to it, or are we
Let
supposed
air
to
get to it? You don't want it going anoxic. So just stand it up somewhere in a safe spot out of direct sunlight and crack the lid so there can be some gas exchange.
Okay.
And it's within 3 to 5 days, really, you may start seeing like some opaque looking, it almost looks like really thin tissue paper on the surface of the liquid.
Did you say it was pink? It should be like pink or something?
No, it'll be kind of white, a hyaline white clear kind of color.
Okay.
But it kind of looks like tissue paper forming.
And then do you want us to take pictures of that too?
Yes. The pollen, when you add water, tends to because hydrostatic forces of that plastic, right? It goes to the edges, kind of rings around the center of your tube. And so that's where your pollen's going to be. Don't keep shaking this up or anything. You want it kind of floating on the top of the water around the edges. And so then you can just go in with some tweezers and kind of scrape the edges where it meets the water, put that on a slide, and you really have a pretty good chance of seeing chytrids after 5 days. Mm-hmm.
Okay.
And what magnification are we looking at?
Whatever works for you. These scopes go up to what, 250? That's going to be plenty to see these things. Yeah.
Okay. Cool.
All right. Other discussion about zygomycetes, things we've read, how they grow.
Dr. Zahn, I've got a
Yes.
question. When trying to review this information for the tests and stuff,
Mhm.
will there be like exact questions from like the specific readings outside of the chapters, basically on everything we've read, right? Or what should we like mostly focus on?
It's on everything we've read and talked about, like in our
Perfect.
lessons. Yeah. But keep in mind it's open note. You have the PowerPoint slides, all that good stuff. So I think you'll be okay. I'm trying to write it more big picture questions than like, what species was mentioned that has a sclerotium underground the size of a football? Like nobody cares.
Okay.
Yeah.
Thank you.
Mm-hmm.
Dr. Zahn, I had a question. I put it in the chat, and we kind of talked about it already.
I don't have the chat pulled up. I apologize.
Oh, it's okay. So we said that the reason it suggested it was more advantageous, the SPK over the ABC, is because more species have it currently. But is there any hypotheses as to why it would be advantageous other than more species have it?
No, and so it's pretty weak evidence, isn't it? It's kind of just a correlation. You can kind of think of it like a botanist saying flowers are advantageous over naked seeds. And if somebody says why, they say, oh, well, ever since the evolution of flowers, like that group has really proliferated and they've kind of taken over the world. And then somebody else may say, well, pine trees seem to be doing fine. And then the botanist says, oh, yeah, but flowers are better. That's kind of what's going on here with the Spitzenkerper. The groups that are doing the best in terms of number of species and diversity do have a Spitzenkerper, but it's not like we're hurting to find zygomycetes. They are doing really well. Every little pinch of soil on Earth has zygomycetes in it. We've even found them in Antarctica. So I think it's kind of a weak statement right now at this point, since we don't have any good tested reasons why a Spitzenkerper would be advantageous over just a vesicular crescent.
All right, thank you.
Mm-hmm. Now, you may find somebody else who has a strong opinion about this and would say, oh, it's obviously better. Send them to me and have them convince me, and I'll change my tune. Okay, from the chapter. Let's see, we had 1, 2, 1, 3, 1, 4. We haven't really talked about we've got the polarity of the hypha. What about cell wall stuff? Secretion, hyphal branching. We looked at the rhizomorphs and sclerotia and all that, but we haven't really discussed the physiology of growing hyphae very much. What do you have to say about that, or what do you think? What about this table right here?
Okay, so I was looking at that a little bit, and the reading was like, hey, this and this, they pretty much just use chitin. But then when you look at the chart, it's only like 50% or 40% chitin in there. So it seems like they are exaggerating it compared to the chart. What's going on with that?
I think you're probably right. It's the classic narrative that plant cell walls are made of cellulose, fungal cell walls are made of chitin, and most people don't get beyond that. But chitin isn't stiff by itself. It can slide all over itself. It's just a sheet of nothing. It's really floppy. It has to be cross-linked by glucans. And so that's what this is showing you here. Let me see, where's the good? Yeah. So this image. So it's showing you the chitin here. And it's showing you that before it's cross-linked, like down here, has integrin proteins stuck through it to help make it more rigid. It's really kind of loosey-goosey, and so that's why the cell is able to grow so well at the tip, and it doesn't explode out the side. So that's what it's getting at there. It's kind of trying to show you that up at the very bleeding edge, there's no cross-linking, and then some starts getting laid down, and it's about 4 micrometers back. Usually it's like already kind of nice and rigid. This is kind of like the party balloon thing, where it just kind of grows out the top because of air pressure. Same thing is happening in fungi, but it's turgor pressure. Somebody refresh everyone on what turgor pressure is. It's been a while for you.
Would that be referring to like turgid versus flaccid, and then turgid is water pressure?
Yeah.
So there's more water pressure on the inside pushing it forward.
Yeah, it's the water. It's the fungus. The fungal cell itself, the hypha, is positively pressurized compared to the environment. So it's a higher pressure system, and that's what's actually making it grow forward. It doesn't have muscles to do that, So it's using water pressure to do its dirty work. It achieves that by having a higher concentration of solutes than the environment. And so this is why fungi don't like to grow in salty areas, because it's harder to have positive water pressure when all your water is rushing out towards the salty water surrounding you. So fungi depend on being in a moist environment because of that. They need water to be coming into their cell to build a pressure gradient, and so that's how they're growing. And so that's what this table is, I think, trying to show, is that it isn't all chitin, right? It's a lot of glucans. And look at this weirdo, Saccharomyces, 1% chitin.
Isn't that because it's single-celled?
Yep, it is. It's a yeast. Yep. Okay, so it's a bunch of glucans, a bunch of protein, and a bunch of lipids. Okay. This one here, Mycota, that's the oomycete that actually shouldn't be in a mycology textbook. And that kind of becomes clear. There's no chitin.
Dr. Zahn,
Yes.
just to clarify, with pseudohyphae and with yeast cells, because that's another type of growth, right?
Mm-hmm.
Unlike septate, where they have pores, is the difference basically that their cells are not sharing their cytoplasm stuff, basically?
Yeah, there's no, in pseudohyphae, it's just a bunch of yeasts that have budded from each other and didn't manage to separate.
Okay, so they're just kind of hanging out, but they're not like in septate hyphae where they actually share stuff. They're just
Yeah.
okay.
Mm-hmm. Our current understanding is that being a yeast growth form is not ancestral for fungi, right?
Okay.
Hyphal growth is the ancestral condition. And so yeast is a derived trait, and they still have all the proteins for like laying down cell walls between hyphae. They just fully close it off, and normally that breaks off. If it doesn't break off, we call it pseudohyphae because it's still sticking together.
Right. Yeah, thank you.
Mm-hmm. It's almost like they want to still be growing filamentous. Yeah. Okay.
Hey, Dr. Zahn.
Yes.
Not to change the topic too much, but I'm wondering if I put too much water on my bread, like when I made the mixture, because I'm still not, I don't know, I'm still not really seeing anything on it.
Nothing?
I mean, I think the bread is just too, like, could you have put too much water? Because the middle of the bread is still, like, wet.
Maybe. They're still going to grow.
I mean,
maybe.
I got started a little late. It's probably only been, like, 4 or 5 days, so it hasn't been as long.
Give it time. Yeah,
Okay.
give it some time. Your bread isn't Wonder Bread, is it?
No, it's just some grainy kind with some seeds on it.
Okay. So it should be okay.
All right. Okay, we'll give it some more time then.
Yeah, maybe open the bag up for a little bit, let it kind of dry out a bit before closing it up. Normally, you can just kind of spray a little water, like a spritz, and that's going to be enough.
Yeah, I definitely doused it a bit more than a spray.
It may be growing just fine. It's just that in the water, the stalks aren't going to be sticking up out of the bread. They're kind of like bound by the surface tension of the water.
Okay. That could be it. Okay. Thank you.
Could you go over real quick the like reduced Golgi apparatus? What's going on with that?
No, because I don't know enough about it.
Okay.
I apologize. Who to talk to? I think Dr. Kudus would be the one to talk to about that.
All right.
I had a question about the slides. So, like between specimens, what's the best way to clean the slide between specimens?
Run it under your kitchen faucet and wipe it off with some paper towel.
Cool.
Yep. It'll be okay. The thing to worry about is the cover slip because they're just so fragile, you know. So same thing with them, but be gentle.
Okay.
Unless you have a large supply of cover slips lying around. Nutrient uptake. Yeah, the ultrastructural cell biology of fungi, I believe, is a bit beyond the scope of this course. So you can kind of skim that. You notice that it's not in my main learning topics. we go. What about AMF? We haven't really talked about them much. Can anyone tell me what AMF stands for? No? Is my microphone on? Can you hear me?
Yeah, it's on. Was it I don't remember exactly what it stands for exactly, but wasn't it the fungi that basically intertwine or have a symbiotic relationship with plants?
Yes. Arbuscular mycorrhizal fungi is the name for them. They used to have their own phylum, but we now know that they tree out within what we call the zygomycetes. So they get their own thing in the zygo group. But they don't make zygospores, which is why we didn't think they were zygomycetes. Okay. The fungus itself is one of the most important fungi on Earth because of the close relationship it has with plant roots. There may be a few hundred species of these, there may be a few thousand. It's kind of hard to tell. They all look identical. Their genomes are really, really big and kind of difficult to study convincingly. But they make those big spores underground. Probably insects or birds are carrying them around. And they're named arbuscular because if this is a type of mycorrhiza, we'll see them again when we look at like fungus plant symbioses. But that little like hand-shaped thing right there inside the plant cell is the arbuscle. Okay, and so the odds are if you see a plant, it's got these living in its roots, and that's the only way the plant is surviving. So what's special about them? Well, they're zygomycetes that don't make zygospores. They are responsible for 80% of land plants being here. They make arbuscles, and they can't live without those plants at this point. So they're also really hard to grow in the lab. You have to actually grow them in a greenhouse with plants to study them. All right, we
and
cool?
They're symbiotic, not parasitic, right?
Yeah, they're mutualistic.
Or mutual. Okay.
Yeah.
That's it.
Okay. All right. I will see you next week, right?
Yeah, we'll see you on Tuesday.
Cool. All right, have a good one.
See you. Thank you.
Do we have anything in between now and next class?
Just work on growing your zygomycetes and add your water to the Chytrid trap.
Okay, cool.
Yeah. So you can upload photos when you're ready to Canvas.
Yeah, I got a quick question. About that bread mold. So I've already taken a bit and put it into the auger.
Okay.
Do we still need more of that bread mold, or we're getting a little heat from my wife right now?
Make sure it's growing on the auger.
Okay. Before I throw away the piece of the moldy bread.
You throw away the bread. Put tape around your auger to like keep it from drying out and keep it from contaminating the rest of your kitchen, and then you're good.
Yeah. Okay.
Yeah.
And we're looking for black mold, right? Not green, because I finally got some stuff, but it's a lot of green and white.
Yeah, we're looking for black. Some of the white might be about to turn black.
It's on top of the green. The
Uh-huh.
white is. Okay,
Yeah.
so
So if the white turns black, then it's sporulating. The fungus itself, zygomycetes, are like clear. In aggregation, it looks white, and they don't go black until the sporangia are being made.
So what if
maybe-
you only? I didn't do this. I got some of the white stuff and put it in my auger. But what if you put some of the green stuff in? Would it still do the same thing, just take longer?
I don't know. What you're setting up there is a battle royale. They're going to be fighting each other.
Okay.
And it's going to be tough to predict ahead of time which one's going to come out on top. So try to go for just the white stuff, or better yet, the black stuff.
Right.
Yeah.
Okay. I'll go from there.
Cool.
Thanks.
All right. See you guys next week.
Yep. Bye.